Dr. Courtney Murren, and I'm from the College of Charleston. And today I'm going to introduce you to some cool tools that we have from Vernier to be able to do laboratory experiments in your classroom on respiration and photosynthesis. So just to orient you, I have the CO2 and O2 sensors already set up in this chamber, and the chamber is really nice because it has these special holes that hold the probes in it. We have just your regular desk lamp, but in this desk lamp we have a full spectrum LED bulb. When we do photosynthesis, we have this water source. It's just an Nalgene bottle filled with regular tap water, and that's so that the leaves don't heat up by the LED bulb. We also have aluminum foil, and as you can see, I've already wrapped the chamber in aluminum foil. Paper towel is just to dry off the leaves should that be needed. And the other tool that we have is a free software from Vernier. And here you can see the icon on my desktop. Let me just open that up. And this is what it looks like, and I'll walk you through it, don't worry. But also on the handout that you have is the location at Vernier where you can download this free software. Okay, now that I've oriented you onto the components that we have available to do these laboratory investigations, let me go over to the refrigerator and get the spinach leaves that I have so that we can start our first experiment. Hi, I just went down to Harris Teeter and bought a bag of baby spinach, and this is to do laboratory 31 from Vernier. So we're going to just take our leaves out, and if any of them are a little bit moist, you want to dab them. And it's probably wise to pick those leaves that are intact, so leave aside the ones that are damaged or already starting to wilt. And what we're going to do is we're going to open up our chamber, maintain that aluminum foil, so you can see the inside of the chamber here. And we're going to just place the leaves to be covering the entire bottom of our chamber. My leaves happen to be pretty dry, which is kind of nice. And these are a little bit cool to the touch, so it may be that the leaves have to equilibrate to room temperature before they start their physiological processes. We're even not going to choose that one, it's a little bit broken. Let's see. A few more leaves out on the table here. Here's a nice one. Maybe it's still a little bit moist. Nice and dry. Place it here in the chamber. Get that covered up. And this one is curly and it wants to jump around. Let's see. I can put it a little bit. Oh, here's a nice one. Okay. Okay. So, close down this chamber now, and you'll want to let your leaves rest for a few minutes. I think Vernier recommends 5 minutes before starting the sensors. So we'll stop the video so you don't have to watch me wait for 5 minutes. But one thing I did want to show you is how these sensors are directly connected to my laptop with these USB cables, and the computer auto detects them. These sensors also, or probes also, can be used through a Wi-Fi setting, and I'll share that those instructions with you as well. Okay, so we're going to wait for these leaves to equilibrate to room temperature, and then we'll start the experiment. Hi, our leaves have now equilibrated to room temperature from after being in the refrigerator. And we're going to open up the Vernier graphical analysis software. And this is what it looks like when I first open it. And there are a couple of things you can do if when you open it, it doesn't look like this. Over here, you can switch to 2 graphs or 1 graph or 3 graphs. You can add the data table if that's what you'd like to see, or if you want to check out what the meters are registering here at the start of our experiment. Okay, so we're going to follow the recommendations of Vernier by going here on this portion to make some changes to the data collection settings. We're going to stick with the time-based mode, and our time units are going to be in seconds. We're going to take 1 sample per second, but our interval is going to be every 15 seconds. And we are going to change the duration to 15 minutes. So we'll have a total of 61 samples in our data table at the end of our run. Okay. The next thing we do is we hit collect. And I'm going to go ahead and hit collect and then pause the video, and we'll return back after our data collection. Hey, our experiment ran reasonably well. Let's take a look at what our data look like. Through the course of the experiment, I clicked on this button here, which allowed me to visualize the entire run of the data. And I actually changed the O2. Originally I had it as percent in the units, but now I've changed it to PPT, so parts per thousand. We can look at the graph now is in the 200s range. So we have the CO2 going up and the O2 going down. Now, we can also click here again and add the data table to our view, and we can scroll through the individual data points. Whoops, not that toggle, this one, here we go. And you can see the individual values for CO2 and O2 over the course of our run. We can also save this experiment, which we'll likely want to do, and we'll do this spinach for 2021. Save that. And in another video, I'll show you about the graphing options, the graphing tools, and how to collect measurements of that students then can compare among runs. All right, now let's uncover our leaves, set up our light system, and switch to our photosynthesis experiment. All right, now we've uncovered our chamber, we've set up our Nalgene bottle for any heat absorption from our LED full spectrum lamp, turned on the lamp, and let's get ready to collect. Same as what we did before for the respiration lab, now for photosynthesis. All right, so now we're going to run that for 15 minutes and join back when we have those results. Hey, our experiment, now with these same leaves that we had for our respiration experiment, now we have the photosynthesis data. And we can see that the O2 does respond as we expect, which is going down and then coming back up through the course of the experiment. And over time, we see that the CO2 is being drawn down due to the photosynthetic action of the plants. Now, if you want to go in and get some important parameters, one might be interested in the slope of that curve. So this tool allows us to obtain that information right from this Vernier graphical analysis. We can click Apply Curve Fit, and linear seems to be reasonable for the data that we have in this experiment. Click on Apply, and here you can see that it has the y equals mx plus b written out for the students, and also then you have that slope which then students can note and compare among experiments. You can also see over here that the data for our data set 1, which was our respiration experiment, and our data set 2, now our photosynthesis experiment, are all saved in that same data frame. So when you download it, you can have both of those experiments together that you might want to look at in different ways. Well, that worked reasonably well. So now let's take a look at a couple plants that are easily obtained here in the Low Country and see what their data might look like as well. Another awesome thing about this particular setup is that you're not restricted to just spinach leaves. You and your students can ask lots of questions without any additional equipment needed. For example, just outside our building, we went to see what kinds of fully expanded leaves were available to us, and we picked some hydrangea leaves. These are commonly grown horticulturally here in the Low Country, and then these beautiful leaves come from a redbud. But you and your students may have a lot of questions about sun leaves versus shade leaves, a fern versus an angiosperm. There's lots and lots of opportunities for different questions and comparisons to be made. So we're going to do a couple runs now of photosynthesis with these two species to see what we get. So now we have our hydrangea leaf in our chamber. We're going to turn on the light and set our graphical analysis to collect and see how different a hydrangea photosynthesis is from what we saw in spinach. Let's go. So we got the results, and the hydrangea photosynthesis results look really nice. Let's take a quick apply the curve fit and grab the slope of this line, and we're going to then come in and compare to what we get from our other species that we've been studying. So now we have our data from the redbuds, and here we're zooming in on the CO2 output. And we can do what we did before and apply our curve fit. Look at that linear curve, and we can see the slope. And what we find is that this is for the redbud is slightly less negative than what we found for the hydrangea. So we can make these kinds of interspecies comparisons to provide understanding about what's going on in the physiological processes of our beautiful plants. So the Vernier graphical analysis has a lot of different options that you might want to explore. I just want to point out a couple things that could be really useful for you and your students. So first is sometimes you might want to start a new experiment and have a completely blank table and start over with your graphs. You might also want to export your data. Both the graphs or a CSV file might be really useful if you want your students to learn how to do means or standard deviations like in Google Sheets, so you can upload the data frame that way. You may also want to just add more data sets to your data, and as we looked at before, the first set that we did was the respiration experiment, and by default it's just called data set 1, but we are able to go in and rename the data set so we can be more descriptive. Like respiration spinach leaves as a rename, and we can see that here, and then that will be retained when we download the data set as a CSV. We can also sit here, rename this data set, photosynthesis spinach leaves, and hold that. You can also delete a data set. Perhaps something went awry, during your run and you didn't want to keep it, you can also delete it. There's a number of different statistics that you can run, means, standard deviations, and you might want to compare a number of different data sets for those kinds of features when you're describing different questions or answering different questions. Okay, so that's just a few more items on what the Vernier graphical analysis could be useful for and adapted for different kinds of experiments that you might be interested in running in your classroom.